Oops. Okay, let me start over. I think I just undid the movie. Let me see here. Nope. Okay, we're still recording. Sorry about that, you guys. All right. So we stopped last time on part 3, which was on amino acids and proteins. We had stopped on slide 42 and we'd asked this question: what determines the amino acid sequence of proteins? What determines the primary structure of proteins? And the answer is the DNA sequence of the protein's gene. So this would be a great exam question, so just as a little heads up. So what we want to try to understand is how the DNA sequence is related to the protein's amino acid sequence. And then we want to explore what happens to the primary sequence of a protein if the DNA sequence of the protein's gene is changed. That is, the DNA is mutated. And there might be a change in the amino acid sequence encoded by that mutated DNA, the mutated gene. And the significance would be if we change the primary structure, if we change the amino acid sequence of the protein, it could change its shape and thus change its function. It might be there'll be a loss of function, possibly a new function might be achieved if the DNA change results in minor amino acid sequence changes. It might be the mutated protein is still functional. So when we get into the microbial genetics unit, we'll talk about mutations and their significance. But again, always, we come back to this little mantra that in biology we think of conformation and structure equaling function. So loss of structure, loss of function, new structure, new function. So the 2 nucleic acids that we'll be exploring, and we'll just introduce basic structure, will be DNA and RNA. So the nucleic acids, which are polymers of nucleotides, the 2 nucleic acids are DNA, deoxyribonucleic acid. DNA is the genetic information of all cells, and we will also remember it's the genetic information of DNA viruses. The second type of nucleic acid is called RNA for ribonucleic acid, and there's many different types of RNA. New ones have been discovered relatively recently. But the 3 we'll focus on are the 3 types of RNA that are involved in the process called translation, where the information encoded in a DNA sequence will be transcribed into an RNA sequence and then eventually translated into a protein. So the 3 RNAs that are involved in converting DNA information into the information of a protein will be ribosomal RNA, and we use a little r here, so we call it rRNA, messenger RNA or mRNA, transfer RNA or tRNA. And just I'll throw this in right now. tRNA is the actual translator molecule of the cell. It's the only molecule that can recognize the alphabet of DNA and can also recognize the alphabet of proteins, and that's amino acids. And we'll come back to that when we get into microbial genetics. And again, these 3 RNAs are involved in the processes we call transcription and translation. And for now, just don't worry about those. We'll cover those in microbial genetics. So all 3 of these RNAs are involved in the complicated process of protein synthesis. In addition, as we mentioned earlier, there's many other there's many other RNAs that are involved in regulation of gene expression. And again, we'll try to hit at least a few of those in the microbial genetics section. Just as we looked at the structure of amino acid, the building blocks of proteins, we want to look at the structure of nucleotides, the building blocks of nucleic acids. So again, nucleic acids are polymers chains of nucleotides. And our nucleotides have 3 basic parts. So they have a 5-carbon sugar called a pentose, P-E-N-T-O-S-E. Pent means 5, ose means sugar. So our pentoses, if I can get my mouse here, there we go. Okay, so the pentose sugars, this is kind of an interesting structure. So in our pentose sugar, we're where my mouse is right now, this would be carbon 1 prime, carbon 2 prime, carbon 3 prime, carbon 4 prime, and this is carbon 5 prime. Now the reason I'm using the prime number is that when we're talking about a nucleotide, I say carbon 1 prime, that tells you I'm talking about a carbon in the sugar, the pentose sugar. In contrast, when we start talking about the nitrogenous base, we'll see that the nitrogenous bases also have carbons. So by convention, when we're numbering the carbons in the nitrogenous base, we just use regular old numbers, carbon 1, carbon 2, carbon 3. Okay, so we have our pentose is part of our nucleotide, and then the second part will be our nitrogenous base. And we can see over here the reason they're called nitrogenous bases is that they contain nitrogen. And we'll be talking about the different types of nitrogenous bases. We find 4 nitrogenous bases in DNA and 4 nitrogenous bases in RNA. And then the third component is a phosphoric acid residue. We'll just call it a phosphate group. And again, we want to remember that those unionized phosphate groups, they ionize, releasing 2 hydrogen ions, so they're very acidic, and that's where the acid of nucleic acid came from. All right, and we will come back and talk about the 2 different pentose sugars, the deoxyribose that we find in DNA and the ribose that we find in RNA. So a real classic short answer question on lecture exam 1 is for you to fill in a table comparing and contrasting nucleotides of DNA and nucleotides of RNA. So let's just do that right now. All right, so if we look at the pentose sugar, in RNA, the pentose sugar we find is called ribose, okay, a 5-carbon sugar called ribose. And what we want to do is take a look at, let's count the carbons here, carbon 1 prime, carbon 2 prime, focus on this hydroxyl group that's attached to carbon 2 prime, okay? Because now we're going to take our ribose, we're going to take off that oxygen, and the result will be deoxyribose. And you can see the only difference is on carbon 2 prime here, there's no oxygen present. So that's deoxyribose. That's the sugar found in DNA. And you might think, well, who cares, right? Well, it turns out, this is really cool, the oxygen on ribose is really reactive. And as a consequence, RNA is chemically less stable than DNA. And so in contrast, deoxyribose, since it contains 1 less oxygen, it's chemically less reactive and therefore it's more stable. Now, one really cool hypothesis is that when the first kind of pre-cells were forming, the first early genetic information may have been RNA. But because RNA is so unstable, it was how should I say this? DNA, through natural selection, chemical natural selection, eventually DNA was chosen as genetic information of cells because it's less chemically reactive, it's more stable. You don't want unstable genetic information because remember, with lots of changes, we end up with mutations, we end up with mutated proteins that can't fold properly, they lose their function, and therefore the cell would die. So we want stable genetic information, and that's why the genetic information of all cells is DNA. Now, in further contrasting RNA to DNA, just real quick quiz you guys, what's the sugar in RNA? It's ribose. And what's the sugar in DNA? It's deoxyribose. Good. Okay. Now let's back up here to our generic nucleotide and let's focus on carbon 1 prime. Carbon 1 prime is where the nitrogenous base will be covalently linked. So let's first focus on the 4 nitrogenous bases found in DNA. So in DNA, we're going to see the nitrogenous base adenine, and the abbreviation is capital A. In DNA, we'll find the nitrogenous base thymine, capital T. In DNA, we'll find the nitrogenous base guanine, capital G. And we'll find the nitrogenous base cytosine, capital C. Now, this will probably be maybe a little bit confusing, but we're going to be talking about complementary base pairing rules. So these bases can form hydrogen bonds, and there's rules that states which base can form hydrogen bonds with the other base. So in DNA, the complementary base pairing rules are that adenine can form 2 hydrogen bonds with thymine, and guanine can form 3 hydrogen bonds with cytosine. And we'll see that's going to be really important in keeping our double-stranded DNA annealed. Okay, now in contrast, if we look at nitrogenous bases in RNA, all we have to remember is the bases are similar, except that in RNA, we do not have thymine. So in RNA, no thymine. Instead, we have uracil. Okay, so if I gave you a nucleic acid sequence, and in one sequence you saw there was thymine present, boom, right off the bat, you know that's DNA. And in another nucleic acid sequence you see uracil, right off the bat, you know that's going to be RNA. The complementary base pairing rules for RNA is that adenine forms 2 hydrogen bonds with uracil, and guanine forms 3 hydrogen bonds with cytosine. Okay. In both DNA and RNA, they will both contain one or more phosphate groups, so that's something that they share in common. This is just an example showing you complementary base pairing, and we can see here's an adenine forming 1-2 hydrogen bonds with thymine. So if this was a lecture exam question, you guys, is this DNA or RNA? And you know because thymine's present, it's going to be DNA. Remember, no thymine in RNA. We'll find uracil in RNA and thymine in DNA. So just a little bit more on complementary base pairing rules. So remember, adenine base pairs with thymine, guanine base pairs with cytosine in DNA. In RNA, adenine base pairs with uracil, guanine base pairs with cytosine. And this is a little bit of detail, but we'll just throw it in there. So in DNA, we're going to see DNA consists of 2 strands of DNA, so we call it double-stranded DNA, and they're held together by those complementary base pairs, those hydrogen bonds right here. So we say the hydrogen bonds formed in DNA are intermolecular hydrogen bonds between 2 separate molecules of DNA. And then we'll see in RNA is a single strand RNA, and it's going to form complex shape based on formation of intramolecular hydrogen bonds. Okay. Now, this is not to worry, you guys. Again, this will be in our microbial genetics section, but we'll see that DNA can also form complementary hydrogen bonds with RNA, and this is what permits the cool process that we call transcription. Okay. In cells, then, DNA will be double-stranded, again held together by the hydrogen bonds between complementary bases. In RNA, this is an example of transfer RNA. We'll see that it's single-stranded RNA, but it forms these complex shape, these complex functional conformation, which are stabilized by these intramolecular hydrogen bonds between complementary bases. So the convention we use, we call chromosomal DNA, the DNA found in our chromosomes, we call it double-stranded or DS, right? And additionally, DNA is helical. It forms like a spiral staircase, which is really quite beautiful. And then again, in our RNA, we have single-stranded RNA, ssRNA, and we have stabilization of the functional conformations by intramolecular hydrogen bonds. This is one thing that could either drive us crazy or keep us humble, in that viruses love to break all of our rules when it comes to nucleic acids. So in some DNA viruses will have single-stranded DNA instead of double-stranded DNA. And in some of our RNA viruses, they'll have double-stranded RNA instead of single-stranded RNA. So the viruses are always keeping us humans really humble. Again, this maybe would help in preparing for the short answer section on lecture exam 1 in comparing DNA to RNA. So guaranteed you'll have this as a short answer question. So remember, the sugar in DNA is deoxyribose. The sugar in RNA is ribose. The nitrogenous bases in DNA are adenine, thymine, guanine, and cytosine. The nitrogenous bases in RNA are adenine, uracil, guanine, and cytosine. Both DNA and RNA have phosphates, and in cells, DNA is double-stranded, and in cells, RNA is usually single-stranded. Again, just showing this beautiful helical structure of the DNA. Oh, and this does remind me, do you recall when we were talking about proteins, polypeptide chains, that we said there was a head and a tail, right? Well, DNA is the same way. So if we took say a piece of chromosomal DNA and we cut across the chromosome, and we looked at one strand of DNA at a time, on one end of one strand, we'd find a free phosphate group. And we'll recall that the phosphate groups, or I don't think I told you this, the phosphate groups are attached to the 5' carbon of our sugars. Okay, so the head, or the start of the DNA strand, is defined as the 5' phosphate end with that free 5' phosphate. Now let's go to the opposite end. Let's kind of follow this guy around. Follow this. We want to stick with the same strand. So down here at the opposite end, we would have a free, what they call a free 3' hydroxyl end. So that's the tail end that attached to carbon 3' is going to be a free hydroxyl group. That's the tail end. So we talk about the 5' end of DNA and the 3' end of DNA. 5' is a head, 3' is a tail. And why that's important is incoming nucleotides can only be added to the 3' tail of a DNA strand. So we say that DNA synthesis is 5' to 3'. And furthermore, what's kind of cool is when we're talking about double-stranded DNA, say of the chromosomes, we have this complementary strand. The reason it's called complementary, let's just walk through the nitrogenous bases. So you can see on this second complementary or sister strand of DNA, the base sequence is complementary to this strand of DNA. So we have thymine complementary to adenine, guanine complementary to cytosine. Here again, thymine complementary to adenine. But note the orientation of the complementary strand. The 3' tail of the complementary sister strand is adjacent to the 5' head of the other strand. And this arrangement is referred to as anti-parallel, an anti-parallel arrangement of the complementary strands of DNA. And again, you're probably like, well, who cares? This later in microbial genetics, when we're talking about DNA synthesis, DNA replication, this is a huge headache for us to obey these 5' to 3' rules, because we'll see that one strand is going to act as a template for synthesis of complementary DNA. And again, that's going to create real headaches when we have to obey this anti-parallel orientation of the strand. So we will definitely be diving into that in more detail in genetics. In this cartoon, if we asked what are the bonds that link the nucleotides together, okay, they're linked through dehydration, synthesis, condensation reactions. There's not like a single chemical bond I can point to and say that's the bond that holds the nucleotides together. Instead, it's a bridge, and I think we have a more detailed picture here. We'll demonstrate that. But the bridges that hold the nucleotides together in a strand of DNA or RNA are called phosphodiester bonds. So again, this is a little bit nicer picture showing the 5' end. This is DNA, there's thymine, right? And I can also see this is deoxyribose. There's no oxygen there on carbon 2'. This is the 5' end, so this is the head of the DNA strand, and here's that carbon 3' free hydroxyl. So this is the tail or the caboose, the 3' hydroxyl end. So again, incoming nucleotides are added to this end only. And just as we saw with our polypeptide chains, our proteins, DNA base sequencing always begins with the nitrogenous base at the 5' end. So if we did the DNA sequence for this strand, it would be adenine, sugar, let's go back, adenine, guanine, thymine, and cytosine. Okay. And here again, showing the anti-parallel nature of the two strands of DNA. Just showing a nucleotide of RNA. So remember two differences with DNA. We have ribose. So this is carbon 1 prime, carbon 2 prime. Notice that there's a hydroxyl group here. There's an extra oxygen there. So this is ribose. Carbon 1 prime, carbon 2 prime, carbon 3 prime, carbon 4 prime, carbon 5 prime. Carbon 5 prime in DNA and RNA both have the covalent linkage to the phosphate. And here's carbon 1 prime. It's on carbon 1 prime that we have covalent linkage to the nitrogenous base. And here's another hint that this is a nucleotide from RNA, because here's the nitrogenous base uracil, which is found only in RNA. This is just a little table that I thought might help you in preparing for that short answer question of comparing and contrasting DNA to RNA. So just thought this might be a little help for you getting ready. And then a unique nucleotide that's found in RNA, not only is it a building block for RNA, but it also has a second really important function, and that is the so-called energy currency of the cell. And this cool nucleotide of RNA is called ATP, for adenosine triphosphate. So it's made up of the sugar is ribose, the nitrogenous base is adenine, and it has 3 phosphate groups. So this is a shorthand for it, adenosine, and then these P's with a circle around it are shorthand for phosphate groups. Now, why do we care, right? Well, it turns out that the covalent bond that links the third phosphate with the second phosphate, when this covalent bond is hydrolyzed, it releases huge amounts of energy that a cell can harvest to do cellular work. And in addition, this second bond here, this also releases a lot of energy when it's hydrolyzed. So with ATP, what we find is that these are kind of like the dollar bills of the energy currency of the cell. So the cell is constantly spending ATP, and it does that by hydrolyzing this bond, releasing energy. The cell captures some of that energy to do cellular work, such as biosynthesis, active transport, motility, right? But because the cell is constantly using up ATP, it has to be constantly synthesizing more, and that takes a lot of energy. So this is the little tilde sign we use to indicate those covalent bonds which upon hydrolysis will release a lot of energy, right here, okay? And again, we know here we have our ribose, and here's our adenine, so adenosine triphosphate. So just as we mentioned, you guys, the cell's constantly spending ATP like dollar bills, right? And this will be a hydrolysis of ATP. So after we introduce the elements of water, we rearrange the electrons, our end products of hydrolysis will be adenosine diphosphate, ADP, right? And then here's that phosphate, that terminal phosphate that got released, our inorganic phosphate, and what the cell was after was that release of energy. In our metabolism unit, we're going to spend hours and hours talking about how the cell can drive this reaction backwards. That is, if it's spending ATP all the time, using it up, then it has to be constantly making more ATP. So we're going to see how the energy finds energy sources to drive the synthesis of ADP. Excuse me, I said that totally wrong. The cell has to find energy to help drive the synthesis of ATP. It takes a lot of energy to link this phosphate onto the ADP. So we'll see all the different energy sources cells use to drive ATP synthesis. So folks, that's the end of our lecture Unit 2, Chemistry, Biochemistry. I think that was part 4, nucleic acids. So I'll stop there. Part 5 will be carbohydrates, and maybe I'll just let carbohydrates and lipids, I'll just make one movie of those. Okay, take care, you guys.